I'm Ruonan Wu, a staff scientist from Pacific Northwest National Lab. So my talk will be a little bit different from the last two talks, talking about the largest viral database, the millions of virus species. We're going to talk about one viral protein, but I promise there will be a very intriguing viral proteins you want to hear about. So would like to share some highlights from our recently published work about atomic level structure characterization of a soil viral auxiliary metabolic gene products. So I will walk you through the discovery path to these beautiful structures as showing up here. Recently, there's increasing numbers of studies also reporting that soil viruses could also carry auxiliary metabolic genes or EMGs. And EMGs are viral version of metabolic genes that are not directly needed for viral replication, but they can redirect the energy and resources in the whole cells or modulate the host metabolisms if they can be expressed and functional. And also the majority of those EMGs detected on soil viruses are found through involved in some carbon-relevant metabolic pathways. For example, in our previous grassland soil viral studies, we found a broad variety of the EMGs that involved in this carbon cycling pathways. And among those, we have detected some key enzymes involved in the chitin decomposition pathway, the chitinase and chitosanase. And one chitosanase were even detected on this high quality complete viral genomes. Then why we're so interested about this chitin decomposition pathway? Because chitin is quite abundant. They are the second most abundant structural polysaccharide after cellulose, and also hydrolyzing chitin or chitosan can provide both carbon and nitrogen sources that can be more available for microbial growth. So if EMGs like chitosanase can be expressed and functional, it may enable the host to access a larger pools of substrate to boost the host metabolisms and maybe in turn benefit the viral survival. Then the key question is, can these viral chitosanase EMGs really be expressed and functional? Then in order to answer those questions, our PNL or SFA team launched a cross-disciplinary collaborations with these national user facilities, including JGI, AMSL, and SSRL. And here's a lot of faces of our collaborators from those institutes. So we try to work together to carefully characterize the enzymatic functions and protein structures of those EMGs of interest. And here we first focus on the glycosylhydrolase family 75 or G75 chitosanase. So first we want to have idea how prevalent those viral chitosanase are, and also how different the viral chitosanase compared to the other microbial versions. So we first search against, of course, the largest viral database, IMGVR, but version 3, and I just know today they have version 4, wonderful job. So we first extract the viral chitosanase sequences from those IMGVR database, and then align together with some bacterial chitosanase and fungal chitosanase, and there's a few archaeal chitosanase from the same GH family. So the viral chitosanase can be detected from diverse ecosystems. So as shown in this tree, we have viral chitosanase detected from aquatic systems that are in blue here, and also some from the terrestrial environment that colored in brown. And there's a few from the engineered environment that colored in orange. It's not really obvious here, but imagine there's some yellow orange dot there. So, so this viral chitosanase actually form these tighter clays separating themselves from the bacteria chitosanase as in pink here, and also the fungal chitosanase that is in green here. So, but like majority of those viral chitosanase actually like also relatively closer to those bacteria chitosanase. So they may suggest they may originate from the bacteria and then modify into this virus-specific versions. But in general, they are phylogenetically distinct from the other microbial versions. But they still conserved the same key residues that were previously identified as the active site of the functional bacteria and fungal chitosanase from the same GH family, 75. And of course, there are some exceptions. There are viral genes. For example, we have some substitutions of cysteine and asparagine in the first conserved residues, which is normally an aspartic acid. in some of the clade 1 and clade 3 viral cytosines. So first, we kind of group those detected viral cytosines in this VEGA clades, 1, 2, 3, and then we subgroup them based on their sequence similarities and also the type of the variants detected on the conserved residues. Then we selected representative sequences that highlight with the asterisks on this tree. And then we share those sequences with JGI. They help us to synthesize them and put on expression vectors so our AMSL staffs could express them and test the enzymatic functions. And currently, we're able to express one viral cytosinase from the clade 3, the deepest place, that is detected from terrestrial, so it's highlighted with these 2 asterisks, that is detected from a terrestrial environment of forest soil. And here shows the viral contig where the studied viral cytosines were detected from. So as you can see, there's a multiple viral hallmark genes, both up and downstream the studied viral cytosines. And we also use a colorimetric assay to evaluate their cytosinase activities. And it's here, it indeed showed a positive endocytosinase activities with optimum pH around 5.1. Then we further purified and crystallized of this viral cytosinase by this thing, so it seems messy. Okay, so like this viral cytosinase actually resolve the structures at SLAC or SSRL, and resulting this ultra high resolutions of better than 0.9 angstrom. So it's mainly composed of these 2 domains. So we have domain 1 here colored in green, composing this central antiparallel twisted beta sheet, and some loops of those domain 1 structures also share structural similarities with some proteins in the PDB. And here's we overlaid 2 top heads on top of the viral cytosin structures. And one is known as the plant defense protein, and the other is the endoglycanase belonging to the GH45 family. But it's not so lucky for the domain 2 that is colored in pink here, which is pretty unusual, lacking those secondary structures and contain this previously undescribed fold. So we called it novel. So then we further leverage this atomic level structure to able us to like locate the conserved residues that were identified from the sequence alignment. So these conserved residues actually sitting at the cleft between these 2 domains. So here is a closer look of those conserved residues. So we have aspartic acid form hydrogen bonds with the other 2, and also have water molecules hydrogen bonded with these conserved residues. Then the next question is this: are these conserved residues related to the active site of this functional viral cytosinase? And again, JGI help us to generate single site directed mutant as aspartic acid and glutamic acid respectively. And indeed, these mutants showed reduced activities comparing to the wild type, which is in green here. Then we further co-crystallize the mutant with the substrate, the chetohexyl, and only the glutamic acid mutant could form complex with the substrate without cleaving the polymer. So again, it's indicate the glutamic acid could involve in the catalytic reactions, and at the same time, it also shows us the substrate could interact with the active site clip forming those hydrogen bonds. So then we try to put together the results here and there and propose a mechanism that the glycoside-bound cleavage by the functional viral cytosinase may have the glutamic acid acting as the catalytic proton donor and aspartic acid acting as the catalytic base accepting protons from a water molecule. So just quickly sum up, this is the very first report of previously undescribed cytosin enzymes encoded via soy viruses with detailed enzymatic function, validation, the structural characterization, and also proposed mechanisms. So we found the viral cytosinase were in general phylogenetically distinct from the other microbial version, although it's relatively closer to the bacterial cytosinase. And the viral cytosinase we detected from the forest soils were found to be enzymatically functional, and also with this unique protein structure containing one of the domains that is previously undescribed. Also, we found the viral cytosines also conserved the same key residues that were previously identified as the active site of functional bacteria and fungal cytosines from the same GH75 family, and also validate as the active site of the viral cytosines as well. And also the co-crystal structures of the single site directed mutants with the substrate shed a light on the latent mechanisms of this functional viral cytosines. And there is another interesting result in the study I want to briefly highlight is the high consistency of the structures that are experimental obtained and AlphaFold predicted. So we have the crystal structure that's considered as experimental obtained that colored in green and pink indicate the two domains that we identified. Also, we have AlphaFold 2 predicted model structures that colored in gray. So they pretty well agree to each other or align to each other, even for those less structured loopy domain 2. And also we further leverage the AlphaFold 2 to predict the GH75 cytosines from the different taxa. So we have the fungal cytosines, bacterial cytosines, and viral cytosines from the major groups that we identified. So just by first glance of those protein structures, it already reflects the differences in the groupings based on the phylogenetic analysis. So all these results actually let us think, how far can we go with AI? And you have heard about AlphaFold in talks that yesterday. So maybe like we could further leverage them to accelerate the EMG discoveries in a relatively more high throughput manner. So if you have a thought about it or want to chat about it, feel free to reach out. Just ignore that line. I didn't realize my talk is scheduled after the poster section. So, but you know how to find me in person at a conference. And also here's my email address. And last, I'd like to thank everyone on the team. It's really delightful, awesome, and close collaborations among this national lab and also national user facilities. And this project is part of our soil microbiome SFA funded by DOE and also supported by our FICUS proposal and SSRL user proposal. So many thanks for those funding agencies as well. Now with that, I'd like to take any questions you may have, and thank you for attention.
Okay, great. We have time for about one question, if anyone had.
Oh,
Hey, Ruonan, yeah,
Hi.
really nice talk. And something that we discussed at your poster yesterday briefly, but I think it's worth kind of bringing up more broadly. So this is really interesting, and this is one protein. And we know we have many, many of them. Do you think that we can scale up this kind of work, or do we need to be very smart and devise some strategy to find, within the millions of proteins, which are the one that we should carefully characterize this way? Like, what path do you think we should take?
Yeah, so I definitely share the same ideas of we have to really validate them and see, and protein structures and enzymatic functions really play important role to determining whether they're really the true proteins there. So indeed, like I mentioned, maybe just speak out of my mind, maybe AlphaFold could help us to screen what type of sequence can fold in a way that is actually potentially functional as enzymes, and then you select representatives and then do all these kind of thing. And also we heard a lot of great talks about a high throughput to validate the genes in a high throughput manners. Probably there are some crosstalks among the teams that are really looking forward to that.